Patrick, thanks for joining me here. Could you say who you are?
Good morning, Hakon. So my name is Patrick Sulem. I'm a medical doctor. I'm educated originally in France, and I'm a medical epidemiologist and public health specialist. I moved to Iceland in 2002. I've been working for deCODE genetics for now 22 years. And if I would have to describe what I do, I'm gene hunting. So I'm going and trying to understand, what are the genes where you have variants that cause or predispose to certain disease, both common diseases and rare diseases.
So within gene hunting, would you say you are specialized in one particular area?
No. So right now what I do is I'm like the chief of the hunters. I try to make sure that all the hunters in cardiovascular, in neurology, in inflammatory disease have all the tools, have all the ammunitions to be able to make findings and to make discovery properly.
Is this an exciting time to be a gene hunter? Particularly exciting time?
So I think what's happening now is that the methodology or the technology exists, which is a sequencing technology. This has just been able for less than 15 years at an affordable price. Only 25 years ago, approximately, the first genome was sequenced. So it's going very fast. Without that, we wouldn't be here together talking. The next thing is...
could I jump in there?
Yeah.
So is sequencing going to become as cheap as a pack of gum, or does it need to be?
You have multiple providers, and the competition is going to become fierce, and the price have to go down. They have been at one time where the price were quite high. There have been always this kind of excuse of not doing sequencing because it was too expensive. Now it's not at prices. It's at prices that are much, much lower than performing an MRI or a CT scan. The problem with maybe sequencing right now is just, so you're doing it for research, you can do it for medical genetics, is how do you perform your sequencing? How do you store data? How do you analyze the raw data? How do you analyze the data? So it's more, how do you take those data and do something useful with it?
In order to do what some people are suggesting and is already being done, newborn screening, how cheap does it have to be? Because then you're just screening, you're just, or newborn sequencing. I mean, you're just sequencing and you're going to put it in a box somewhere. So maybe it has to be as cheap as blood type. Is that true?
I'm going to try to answer your question in a few steps. A few years ago, we were at $1,000 genome, or people were hoping for $1,000 genomes that have been reached. And then after a few years, they say, we want to go to the $100 genome. And that's what we're aiming now. It's going to be in the next few months, few years. We're hoping for that type of price to be able to perform a lot of sequencing. The thing is, when you're talking about going much lower, I think, there are some technologies that might allow this type of price to be reached, maybe in the tens of dollars. But right now already, we are at a time where it's affordable. You can do it for kids that have rare disease. You can do it in research. It's just a matter of scale. It used to be in the tens of thousands of dollars, the genome, when it started, maybe 15 years ago. So I think we're getting at places where you can perform whole genome sequencing.
So we're at this conference, the Nordic Conference on Future Health. First time we brought together the biobank and research communities, although many biobankers are researchers as well. Is this a good idea? Do you think that to bring these people together?
It's very helpful because, so as a researcher, what you want to do is, in general, find collaborators. But very often the collaborators are not necessarily the people that are storing the sample, are not necessarily the people that are storing the information. So here we have both types of people, the people that are working with data, the people that generate the data, that store the data. So I think it's a good mix.
Anyone missing from this equation right now?
Right now, no. I think it's a very good mix. I remember a few years back, we were together also in Norway, in Oslo. I think it was more focused on Biobank. And I know my colleagues has gone to some Nordic conference where it was more focused on research. I think it is a balance now.
What about clinicians?
The thing is, I think it's always good to have some clinicians, but it's good to meet clinicians at certain type of conference. You do meet them when you go to the large genetic conferences, but it's just on genetics. They have in general a very specific idea of what they want to get out of the research knowledge, and it's often an action or a practice change. They want to know what they're going to do. Some clinicians are also researchers, but in that case, they're probably here.
And so beyond just having a meeting, I heard you say that we need involve the clinical communities.
Yes.
And can you say more about what their role there is?
So I think there's probably a double role. First role, they will be users of the data that will be derived from research. So the researchers have to think about how the data they're generating can be used in the future. And we've been discussing about actionable genotype. We've been discussing about polygenic risk score. All those things are only useful if they go into the medical field. And once they go in the medical field, the medical doctors and the medical community will have to be getting used to this type of information. The second thing is you cannot do genetic research without good phenotypic or medical information. So at the same time that the medical community is going to be using the data derived from research, they're also feeding, and in fact, a lot of researchers are medical doctors or are heading in medical groups. So the medical information is also what is the prerequisite to perform genetic study. So you need to be able to compare genetic polymorphism to phenotypic polymorphism.
So in this year's meeting, have you seen or heard anything that you found particularly interesting?
So the first thing that we've done is to contact groups that we didn't know about. Some people, for example, working on different materials than the one that we already have been contacting us. We're going to have lunch today with a cancer group from Sweden, and we're going to discuss the type of material they have, what they want to do in the future. We're going to compare with what we have done. So it's something that is quite useful. Just discover new players in the field, or players that are new to us, not necessarily new in the field, but new to us. The second thing is we are hearing a lot that people want to work together. So at deCODE Genetics in Iceland, we have been collaborating heavily with the different centers in Norway, in Denmark, in Sweden. We are also in communication with the Finnish groups. But definitely, it sounds like people want to work more together. They want to be able to benefit from the experience of each other. And it seems that right now, most of the effort are still national efforts, a lot of them. So I hear that, and it's very interesting for us because we've been doing collaboration, and we're extremely happy to hear that people want to collaborate in general.
In order to bring together data at a Nordic level that are similar and that can be worked on together, much more easily than data from other places where you don't have the same depth of phenotypic information. What are some of the things that you heard in the session earlier today, or that you think need to happen to create this?
So one thing that is clear is when you're thinking about disease, so now we're going to go on two different levels, the phenotypic level first and the genotypic level. You need to have standard. You need to agree on certain way of communicating. So when it comes to disease, if I tell you, I have 10 patients with asthma, and you tell me I also have 10 patients with asthma, they might not be exactly the same. How do you define your phenotype? How do you define your disease? Is it based on the hospital? Is it based on the general practitioner diagnosis? Which classification did you use? Which code did you include? So some of the groups here have already been trying to do algorithm or recipes for each of the disease. And I think that is something people will just probably have to try to agree on what they use. So in order to not to have to reinvent the wheel and reinvent the definition of each of the disease. So if we would talk now and you say, I have a cohort with asthmatic patient, I also have a cohort with asthmatic patient, it would be very good to know according to which standard. So I think the phenotype needs to be somewhat standardized, and the definition needs to be standardized at the level of the different research center. And you're always going to keep the raw data, but you probably want to be able to have something that is something that you can communicate with a common language. when it comes to genotype, it's going to be probably important to realize that here in the Nordic countries, there is a lot of commonalities. There is a lot of sharing of population history. And very likely, one thing that is important on the genotypic level is a rare variant does probably not care about boundaries of frontiers and might be somewhere in Norway, somewhere in Sweden, somewhere in Finland, because there are some populations that are going over those three areas. And I think it's very important to keep that in mind, that whatever on genotype you will learn about one population, it will help the other population. So at deCODE, we have genotypic information. And one thing that we realize is you often want to process it in a similar way. You often want to be able to have things done in a homogeneous manner when it comes to sequencing. And these things, those are just protocol that needs to be standardized. And for example, what technology people are going to use. Is it going to be whole genome? Is it going to be short read, long reads, whole exome? I don't like much whole exome, so I like to focus on whole genome. I think it's a kind of what's going to be used mainly in the future, because people are realizing now that they are, even on the rare disease, there are solutions being found there. So I think the whole genome will become the standard. But then it's just, how do you do it? Which steps do you use? So I think a common language for phenotype, a common language and protocol for genotype, that you want to kind of be able to work together. So I think the commonality of language for those two things is very important.
Everyone is familiar with the figure that you may have shown. Everyone shows the Terry Manolio.
Common variant, rare variant.
How relatively important are they for disease?
So we're looking into all of that, but right now what we have to keep in mind is most people that are studying genetics have almost a hard cut of around 1%. So they're not really looking very deeply there. And there's two reasons for that. The first one is that most people are using genotyping array, where they later on impute genotype that is coming from another data set very often. And so the level of information that people have below 1% is in general bad, is very often bad. Whereas if you do whole genome sequencing, or if you do a local set of imputation with a large fraction of the population with whole genome sequencing that you impute to the rest, you can get better. But historically, people have kind of stopped around 1%, things above 1% they're sharing, below they don't completely trust it. So most of the genome-wide association consortium have result only on variant over 1%. So a lot of the things that are below that are not really well understood right now. Definitely, there are variants that we're finding there, and the reason why you find them with a large effect, the rare variant, is because if they would have a small effect, you wouldn't see them. So they're also going to be rare variant with a small effect, but you cannot provide evidence for those. So right now, for common variant and large effects, those things have been discovered a long time ago, like the apolipoprotein E for Alzheimer, a lot of the HLA type for autoimmune disease. So we're looking for this low frequency to rare variant with large effect. Individually, if they have a large effect, they have the potential to have an information that is very relevant for the individual. So we are going closer and closer to monogenic trait, or to things that are more deterministic. So it might have a very high impact on the individual level, even if the impact on the population overall might be larger for the common variant with small effect.
Right. So 2 years from now, we're going to meet in Copenhagen. What would you like to see at that meeting in Copenhagen? The either a development from what you've seen here, or something that was missing?
I would like to see a large set of whole genome sequence in each of the Nordic country. I think this would enable the research, the genetic association research. This will enable the medical genetic, and this will definitely be something that will happen at one time. Is just the earlier, the better.
Do you have a bold statement that you might like to make about an important, or maybe even controversial aspect of this work and this future of precision healthcare? Just, something you think you might say. You might admit you're wrong in 5 years, but right now it seems like something to say.
So genetic research is not becoming about doing your own research. It's also about being on the watch for whatever is available publicly. So trying to take the benefit of all the groups that have put data public. We have contributed, we have at deCODE Genetics, whole genome sequence, half of the UK Biobank, and those data are enabling a lot of research. We are also ourselves benefiting a lot from data that have been made public, like the All of Us data in the US are becoming available to more and more researchers. The Million Veteran Project has released their summary statistic recently. So there are more and more projects that are putting to one level or another, either the individual level data or the summary association data public. And that really is something that is going to be, I think, the game changer. Is you will have not only to do your own research, but to add your research to the one that are publicly available. So we are now, for example, at deCODE Genetics working with up to 18 population, and it's becoming now close to half of this data that are public data that are coming from very many different research groups in Europe, in Asia, in the US. So becoming a good bioinformatician is necessary now. You need to download this data. You need to curate them to a level that you make them homogeneous with your own data. And I'm often talking about just summary statistic, but it's becoming really a difference because 10 years ago, we're thinking about how we're going to generate our own data. We've done it. So now it's how we're going to add our own knowledge to the knowledge that are public and that are becoming larger and larger. And just finding all these sources is a challenge.
That's a promising future. Where people make data available on a huge scale like this publicly, and 1,000 flowers of bioinformatics bloom.
And not all of them are to the same level. Some of them might be focused on the disease association to genetic. Some of them might be understanding the transcript. So you just have to combine not only one type of data, but different layers, the transcript, the protein, the genetic association, and you have to try to put them together. And we're not even going into all this annotation of the genome itself. That's even another task to tackle in the future.
Well, great. Thank you, Patrick, for joining us.
Thank you so much, Hakon.
I'll see you in 2 years in Copenhagen.
See you in 2026.